talk about here is the regional specialization of the renal tubule. So we're going to talk about how different parts of the tubule look differently and have different functions, okay? And so the main three distinctions that we're going to talk about are the proximal tubule, where we see unregulated or non-regulated reabsorption, the distal tubule and the collecting duct. We usually talk about these two areas together because the features are kind of the same. Here we see regulated reabsorption and secretion. So we don't see any secretion in the proximal tubule, but here in the distal tubule, we do see secretion along with regulated reabsorption. And then we're going to talk about the loop of Henle, where we see water conservation. So water conservation, meaning that we allow the body to hold on to more water. And this is where we really concentrate the urine and make the urine less watery. This mostly happens in the loop of Henle. We're going to talk about that as well. So this is a really good table for you to use as a study tool, to test yourself, to make sure that you understand what's happening at the different parts of the tubule. It's going to be a really important kind of big takeaway message here. I urge you to also create another column where you mention some of the anatomical features. So we're going to talk about the different anatomical features that are seen in the proximal tubule, different features that are seen in the distal tubule, collecting duct, some of the different cells that are named differently in the distal parts of the tubule. So I encourage you to create another table, another column here, and mention some of the anatomical differences, like the types of tight junctions that are different and so on, and try to recreate this table as a study tool. So in the proximal tubule, we see most reabsorption happening. So that's the general trend. I don't really want you to memorize what's happening, but try to understand the proportions and the general trend that's happening. So most of the reabsorption is happening in the proximal tubule. Most of the reabsorption of water is happening in the descending limb of the loop of Henle. So that's the first part of the loop that's going down. Most of the solute reabsorption is taking place in the ascending limb of the loop of Henle. So that's the part of the loop that's curving back up. And so we're going to talk a lot about how the descending limb is reabsorbing water and the ascending limb is reabsorbing solute. So you're going to want to make a note of that. The distal parts of the tubule, we see regulated reabsorption happening of water, and then we see some secretion happening as well of sodium, of potassium, and of hydrogen ions. Alrighty. Okay. So let's talk about the proximal tubule and how reabsorption happens here. So we know that this is the mass reabsorber, and 70% of the sodium and water is reabsorbed here, and 100% of the glucose is reabsorbed here as well. Now the cells of the proximal tubule look very different to the cells of the other parts of the tubule. They have certain modifications that let us know that they are adapted for reabsorption. The first one is called the brush border. So the brush border is this highly folded apical membrane of the epithelial cells of the proximal tubule. And it's also studded with microvilli. So the microvilli and this highly folded membrane is actually increasing the surface area of the proximal tubule and just allowing more space for reabsorption. The second modification is the leaky tight junctions. So in between each of these epithelial cells, there are tight junctions that are kind of anchoring these two cells together, but the cells of the proximal tubule have leaky tight junctions, meaning that they do allow some movement of substances in between the sides of these cells. So substances that are going from the lumen of the tubule out into the peritubular space and peritubular capillaries can actually be moved in between the sides of these cells in order to be reabsorbed. So the leaky tight junctions along with the microvilli and the brush border are some of the modifications that let us know that this is really where lots of reabsorption take place. And when we talk about the gastrointestinal system, we're going to see this exact same modification, which again is an indicator of the mass amounts of reabsorption of nutrients that happens along the GI tract, along the small intestines. So here's a kind of close-up of what this looks like. We have leaky tight junctions here. We have the extensive microvilli, which is forming this highly folded brush border. And when we say brush border, it kind of looks like a bristle brush because of all of these, you know, spiky microvilli and the highly folded nature of the apical surface. And again, I'd urge you to put these features in that column that I talked about in that table. Okay, let's look at the cells of the distal tubule and the collecting duct. And again, we talk about these two areas of the tubule together because the features are kind of the same and the mechanisms are kind of the same here as well. So here we see regulated reabsorption, meaning that hormones are going to influence the reabsorption here. Your hydration status is going to influence the absorption here. So for example, there are certain hormones that are going to increase how much water is reabsorbed and certain hormones that are going to decrease how much reabsorption is taking place. And that is what we mean by this area being regulated. It's going to happen under a very tight or very controlled way or very controlled mechanism. And so another thing that we see here, which tells us that there's regulated reabsorption, is the tight junctions. So there's very little paracellular movement of substances and water happening in this part of the tubule. So substances cannot move in between the sides of those cells. And again, the reabsorption here is very heavily controlled or regulated. Another thing is that we don't see the microvilli. As we can see here, the cells are more of a cuboidal shape. They don't have those microvilli. They don't have the increased surface area, which is another feature indicating the unregulated and free reabsorption. Now later on, we're going to talk about some of the names of the cells here, but we have two types of cells that are present in the distal tubule and collecting duct. They're called intercalated cells and principal cells. But I'd urge you to make a note on this slide because we're going to come back and I want you to kind of remember that these are the same types of cells that we're talking about when we point them out later on. Okay, now the loop of Henle. So the loop of Henle is that part of the nephron that dips down into the medulla and then dips back up into the cortex. And the loop of Henle is really important for concentrating the urine, meaning that it's going to minimize the amount of water that we lose in the urine. So the loop of Henle is very important, especially in animals that have limited access to water. So think about camels. Think about animals like desert rats that go for long periods of time without water. They have really, really long loops of Henle. Humans have a somewhat long loop of Henle as well. And so it's going to allow us to go for extended, not extended, but somewhat extended periods of time without water. And the kidneys, specifically the loop, can concentrate the urine and prevent us from losing a lot of water because it is giving more space, more time for water to be reabsorbed and for the concentration of urine to take place. Okay, we're going to talk all about the mechanisms that happen or the mechanisms that are controlling the reabsorption of water in the loop. We'll talk about how the descending limb is different from the ascending limb in our next packet, but we really want to just point out here that the concentration of urine is what takes place. Okay, now we're going to move on to talk about a few terms here. We're going to talk about excretion rate, which is the rate at which products or substances are cleared from are excreted from the body. We're going to talk about clearance. And then finally, we're going to talk about micturition, which is the process of urination. Okay, so the excretion rate is basically the amount of substance or the rate at which an amount of substance is being excreted from the body. Now we know that how much is excreted is equal to the amount that's filtered across the glomerulus. We're going to add the amount that's secreted. Remember, secretion is movement of substance and substances from the peritubular capillaries back into the tubule. So we're going to add that. And then we're going to subtract the amount that's reabsorbed, right? Reabsorption is the movement of substances from the tubule into the capillary. So we're going to add what is secreted and subtract what is reabsorbed, and that is going to give us the total amount that is excreted. The excretion amount is going to depend on how much was filtered to begin with, so the filtered load, the rate at which secretion is taking place, so how fast substances are moving from the peritubular capillaries back into the tubule, and then the rate at which reabsorption is taking place, so the rate at which substances are being moved from the tubule back into the peritubular capillary. And so from that, we can deduce that if the amount of solute excreted is less than the amount that was filtered, then solute must have been reabsorbed. We can also deduce that if the amount of solute that is excreted is greater than the filtered load, then solute must have been secreted, okay? So take a second and make sure you really understand that and kind of let that sink in. So if the amount of solute is excreted is less than how much was filtered, then it must have gone somewhere, right? And so we say that it was reabsorbed back into the blood. But if it's greater, the amount of solute excreted is greater than what was filtered, then it must have come from somewhere. And so solute was added or secreted from the capillaries into the tubule. Alrighty, so if we compare the osmolarity and the total volume of the solute, when it's first filtered, the total osmolarity is 12 millimoles. And this is just a representation of how much solute is dissolved in the filtrate at this point. And the total volume is 150 of water. The total volume of water is 150 milliliters. Now, as we move through parts of the tubule, we see that 3 millimoles of solute is secreted into the tubule. And so that gives us a solute concentration of 50 millimoles. And we see that 9 millimoles of solute is reabsorbed back into the capillaries, and 145 milliliters of water is also reabsorbed back into the capillaries. So at the end of the tubule, we have a solute concentration of 9 millimole and a water volume of 5 milliliters of water, okay? And so that is telling us the movement of solutes and water across the tubule. That is giving us an idea of how much is secreted and how much was reabsorbed. Now, this table is giving us an idea of some of the clearance rates of common substances. And I don't really want you to memorize this again, but I do want you to have an idea of understanding what clearance means based upon these values. So PAH is para-immunohepatic acid. This is actually an exogenous substance that's put into the body, that's injected into the veins, and we use it to measure the clearance rate because it's secreted. Whereas something like glucose is filtered, but it is fully reabsorbed, and so it has a very low clearance rate, right? It should be 0, meaning that we should not see any glucose coming into the urine, whereas we should see a lot of this PAH coming out in the urine, right? Because it's going to be filtered and secreted from the blood. And so the clearance rate is basically a measure of how much that product is coming out in the urine, how much that substance or that breakdown product, in the case of creatinine, is a breakdown product of muscle, how much that is actually being cleared in the urine. And what we can deduce, the relationship that I want you to deduce here, is that if the clearance is equal to the GFR, then the substance is neither secreted nor reabsorbed. And that is the case with inulin. So inulin has a clearance rate that is the exact same as our GFR. And so this means that the exact amount that was filtered is the exact amount that's coming out in the urine, and the same rate, 125 milliliters per minute. Whereas if we look at something like glucose, if the clearance rate is less than the GFR, right, if it's 0, that means that net reabsorption takes place, or all of that product was reabsorbed in the case of glucose. So 0 of it is coming out in the urine, right? 0 milliliters per minute. Or we can look at something like creatinine. If it's greater than the GFR, if the clearance of the substance is greater than the GFR, the net secretion must have taken place. Because if it's coming out at a rate that is faster than the rate that it was being filtered at, then it must be coming from somewhere else. And we can say that it's being secreted from the capillaries into the tubules. All right, so the main takeaway that I want you to get here is that if the clearance is greater than the GFR, net secretion is taking place, and if the clearance is less than the GFR, net reabsorption is taking place. If the clearance is 0, then 100% reabsorption is taking place, and if the clearance is the same as GFR, then neither secretion nor reabsorption is taking place. All right. Okay, let's pause for a minute. Let me see if there are any questions on any of that before we move on to our fourth objective. Okay, I don't see any questions. So let's move on here to look at micturition. So micturition is basically the process of urination or voiding of the bladder. It's basically emptying of the urine that is being stored in the bladder. So remember, the kidneys are making urine. It's being transported to the bladder by the ureters. So we have 2 ureters, one coming from each kidney, excuse me. And that is bringing urine to the bladder. And then in order to void that bladder, so the bladder is just storing urine, in order to void that bladder, the urine must leave the bladder and enter the urethra. So we have 1 urethra, but 2 ureters, and be eliminated from the body by the external urethral orifice, okay? And so the purpose of the bladder is kind of to store urine so that we're not urinating every 5 minutes as urine is being constantly created. So the detrusor muscle is the muscle that lines the wall of the urinary bladder. It is smooth muscle, so it's not under our voluntary control. It is extensively connected via gap junctions, and it's innervated by the autonomic nervous system. So it's going to be innervated by the parasympathetic, which is one of the divisions of the autonomic nervous system that's important for voiding of the urine. There's also sphincters that are around the urethra. So the urethra is the tract that is exiting the bladder and leading to the external orifice or exiting the body. And it's guarded, or it's surrounded by 2 sphincters. One is the internal urethral sphincter, which is closer to the neck of the bladder. This is formed of smooth muscle, and so this is under control of the autonomic nervous system. And then there's the external urethral sphincter, which is closer to the exit, closer to the external orifice. And that is skeletal muscle, which we voluntarily control. So the internal urethral sphincter, smooth muscle, under control of the autonomic nervous system. The external urethral sphincter, skeletal muscle, under control of the somatic nervous system. Now, typically when the stretch receptors in the bladder detect that they're stretching in the wall of the bladder, meaning that the volume of urine is building up, it'll send information to the regions of the spinal cord. So that's S2 to S4, the sacral regions of the spinal cord. And the neurons here are typically inhibiting the parasympathetic response. So the parasympathetic is what's going to cause you to void your bladder. But when you're moving around during your everyday life, you don't want to be continually, sending that signal to void your bladder. So we're inhibiting the parasympathetic nerves to the muscle and keeping the external urethral sphincter stimulated. This is called the guarding reflex, and this is what prevents you from voiding your bladder as you're kind of moving around in everyday life, just involuntarily. But when you are ready to void your bladder, this other reflex that is elicited is called the voiding reflex. And so instead of inhibiting the parasympathetic neurons, we stimulate the parasympathetic neurons. This causes the detrusor muscle to rhythmically contract, and then we inhibit the sympathetic innervation of the internal urethral sphincter. This causes it to relax, and then obviously the person is going to voluntarily decide to go to the bathroom, which relaxes the external urethral sphincter. Okay, so there are really three different divisions of the nervous system that are happening in order for you to void the urine and go to the bathroom. You have to inhibit inhibit the sympathetic, which allows your internal sphincter to relax. You have to excite the parasympathetic, which causes the detrusor muscle to rhythmically contract. And then voluntarily, according to the somatic nervous system, you have to decide to relax the external urethral sphincter, and that allows you to void the bladder. Okay, and here's a flow chart that kind of lets that make a little bit more sense. So the bladder is full, the volume has increased, and there's stretching on the walls of the bladder. This is going to activate the stretch receptors in the wall, and that is going to send signals to areas in the brain, which is going to decrease the sympathetic activity, facilitating the relaxation of the internal sphincter, that sphincter that's closer to the opening of the bladder. There's also going to be simultaneously an increase in the parasympathetic activity that causes the contraction of the detrusor muscle itself. Remember, that was otherwise being inhibited so that you didn't use the bathroom involuntarily. And so both of these signals allow the opening of the internal urethral sphincter and the contraction of the bladder. And that's kind of one part of the voiding. The third part is that you have to voluntarily decide via the somatic nervous system to relax your external urethral sphincter, and that will allow urine to leave the body through that other sphincter closer to the external orifice. Okay, so these are the three parts to the voiding reflex that allow you to void the bladder. Okay, so here's another set of checkpoint questions. Again, similarly, we're going to look at these the next time